Welcome to the Uncommon Living podcast. I'm Thomas Seeger. I'm an associate professor in the School of Sustainable Engineering at Arizona State University and the CEO of the Morozko Forge Ice Bath Company. My guest today is Kristy Townsend, who is a professor of neurobiology at The Ohio State University. In 1 hour, Professor Townsend corrects decades of misconceptions about the role of fat inside our body and the way it interacts with our nervous system. Kristy Townsend, thank you for joining me on the Uncommon Living podcast.
Great to be here. Thanks for having me.
If I was going to write a book called The Secret Life of Brown Fat Cells, I think you would be the main character.
I'm not sure about that. I definitely did a lot of work in brown fat in my career, but now I think the title might be The Nerves That Innervate Our Fat Tissue. That's more my niche these days, but that relates to brown fat and white fat and all of our tissues and organs that matter for metabolic health.
Our popular understanding, at least in the United States, is that fat is bad, that fat is an energy storage organ. And of course, it is associated with several adverse health outcomes, heart disease, even dementia. And so everyone that I know, including me, wants to shed a few pounds. We're heading into the holiday season, and it's only going to get worse. But your work is revealing that fat has a much more complex relationship with the body and the brain than we've been led to believe. I'm hoping that you can give our audience a little bit of a primer on what they need to know about the nervous system and adipose tissue.
Great. Yes. So adipose tissue is the medical and scientific term for our fat tissue. So we can use either word today. And it is a tissue that gets vilified a lot, I think. So we hear about having too much unhealthy fat tissue in the body, and that leads to obesity and then additional diseases like diabetes, cardiovascular disease. Obesity is also linked to some cancers. But in fact, it's not just having the fat tissue that's causing these health problems. It's where the fat is in the body, what type it is, and how healthy it is. So as you alluded to, we have white fat and brown fat. Those are the two major types. The majority of fat in our body is white fat. And that the job of that is to store fuel for us. So it stores fats, triglycerides from our diet and from other sources around the body. And it's there when we need it. So if we're going to run a marathon or climb a mountain or go through a period of fasting, we need that fuel stored. So it really is an evolutionary advantage that our fat tissue can do that. Brown fat, on the other hand, evolved to keep us warm. So babies and rodents and things that need to, they're small and they need to produce their own heat have this special brown fat. It also stores fuel. It stores triglycerides as well, but it can use it to make heat and burn calories. And that's why we think of brown fat as sort of the healthier or healthiest of the fat tissues in the body, because it's doing a lot of things that prevent obesity and prevent diabetes. It's taking up and burning a lot of fat around the body, taking up and using glucose and sugar. So it's a healthy fat to have. But that being said, our white fat is not all bad either. We have fat under the skin, subcutaneous, and also around our visceral organs. And it's really the fat under our skin that's healthier. It's more insulin sensitive. It's secreting different hormones in the body that we need. So typically when people are looking at someone who's overweight or obese and they're trying to figure out if they might be at risk for future metabolic disease, they're looking at their waist circumference. They're looking at their visceral fat around the internal organs, because that's really what will predict. Subcutaneous fat is protective in some ways, but that changes as we age. And I think we'll talk about that. So as we age or as women go through menopause, those depots of fat and how healthy they are can shift. But what I've given is sort of the broad strokes overview.
Then let me see if I can summarize this correctly. So far, you've mentioned 3 types of fat. There's white fat, and that comes in 2 forms, subcutaneous and visceral. The
Yes.
subcutaneous is fairly benign, at least compared to visceral. The subcutaneous is what's right under the skin. When I was a kid, I think Special K used to have a commercial about pinching an inch. But that was subcutaneous fat that I'm pinching on my belly. And although it doesn't look good and it doesn't feel good, you're saying it is nowhere near as dangerous as the visceral fat. The visceral fat is around the internal organs, and this is what's associated with adverse health outcomes. Then
That's right.
there's, all right, then there's brown fat. And brown fat is located in different places in the body. You can't really see brown fat. It's much smaller in quantity, and it has almost this opposite function. Although brown fat is still a fat, it burns up calories, so to speak. That is, its job is to keep us warm, not as a source of fuel, but as an engine of thermogenesis. That is, it would be associated with healthy outcomes, improved insulin sensitivity, leaner body composition, and longer life, which is the opposite of the visceral fat. Now, I think it's confusing, but when we sort it out that way, three different types, one of them healthy, one of them unhealthy, and the one in the middle, the subcutaneous white fat, sort of benign. Am I getting that wrong?
In broad strokes, yes, absolutely. Yes. And brown fat, so in humans and adult humans, it is hard to see where it is. That's right. There's a lot around our collarbones and our spine, kidney, or a little bit around the kidney and heart. But if you are around a baby and you put your hand behind the shoulder blades of a baby and you feel a lot of heat coming from there, that's what we call the interscapular brown fat pad. And that disappears as we age. So around 7 years old, that starts to atrophy. But that's probably the biggest brown fat depot we have in our lives. And babies need it to stay warm. So that's the one interaction with brown fat you sort of can really experience in your life. And I'll also add quickly that, fat tissue is specialized to store fuel. It's specialized to store these triglyceride fats for when we need it. But what really causes these metabolic diseases is when we have too much to store properly and then it spills over and we start to get fat in other tissues and organs, fatty heart, fatty liver, fatty muscle, fatty pancreas even. That's really what's dangerous because those tissues and organs are not built to store fat. And when they need to, because we have too much and the capacity of our adipose is overcome, that's really when we get problems systemically.
I think that makes sense. You're saying it is when our subcutaneous fat or our capacity to store that energy in the subcutaneous fat is overwhelmed and we begin to pack fat into other places, white fat, where it doesn't really belong. And that's when it becomes dangerous.
That's right.
I was talking with Stephen Cunnane at the University of Sherbrooke, and Stephen has this really smart, convincing approach to understanding brain development and its reliance on DHA, on AA, and the way that human beings must be getting this from aquatic, from fish, from plants living in the water, from aquatic sources. We talked a little bit about what Hardy called the aquatic ape hypothesis, which points out that humans have subcutaneous fat. But Kristy, why don't the apes have it? Why don't bonobos and chimpanzees and silver? If subcutaneous fat is good for energy storage, why are we, the creatures that have it when these other land mammals just don't seem to use it?
Well, I don't know how much that's true. I definitely don't work with primates, but in the laboratory, when we work with preclinical mouse models, rodents, they do have subcutaneous fat. So I'm not sure if we're really that unique. We use a lot of model species that have similar fat depots to us. They have brown fat, they have subcutaneous white, and they have visceral. So I'm not sure. I can't really speak to apes, but the rodents have it. So it's stuck around in evolution.
A lot of the work in fat, in metabolism, and just investigation of fat is done in rodents. These convenient rodents with their brown fat and their short lifetimes allow us to, investigate hypotheses and get a rapid return. But there are some limitations to the way that a rodent model will port over to human beings. Denis Blondet, also at the University of Sherbrooke, he was the one who pointed out to me that rodents have receptors in their brown fat that are absent from human beings. And so a rodent brown fat might be activated sort of pharmacologically or something in their diet in ways that humans just don't respond to because we lack that receptor. Where should we be cautious in
Yes.
interpreting those rodent models and how they might transfer?
Yes. So first of all, I want to emphasize that we can't get around in medicine working with model species. We just can't. So, some of the rhetoric right now about, just using organoids, just using cell culture and then moving to human. I have no interest in taking a drug that was developed that way. So I think we have to look at the appropriate preclinical model, whether that's a mouse, a rat, what have you. They are, for the most part, very similar physiologically and genetically to us. Now, there are exceptions, and we do need to pay close attention to those, and we do need to be very rigorous in our research and make sure that an observation in a mouse can translate to human. So for us, we do work mechanistically in a mouse model. It gets diabetes after obesity. It gets a lot of the complications like neuropathies that we get as humans. So for us, it's a largely good model. But exactly, there are differences molecularly. There are differences in immune systems. There are some differences in metabolism. So rodents rely a lot more on thermogenesis than we do as humans, where we're in heated homes and with clothing, and we have larger bodies that are storing more insulating fat. So we have to be cautious. But the goal with translational research is to start in a model species, learn mechanism, get some hints at potential, targets, and then we have to validate in human and make sure that what we're looking at is actually translating to human health.
One of the things that I've read about brown fat is that it is highly innervated. That is, to activate this thermogenic response. Sometimes your body wants to do it fast. I get into the ice bath and my body immediately understands that I'm in thermal stress. And so it's going to release glycogen from my liver to boost my blood glucose, and that will fuel the mitochondria in my brown fat and my muscles so they can produce heat. And for this to happen fast, it's got to happen through the nervous system. White fat, less innervated. Will you tell us about what you're learning on the relationship between the nervous system and the fat, all the different types?
Yes. So one of our main interests in my lab, I am trained as a neuroscientist, but we're interested in how our fat tissues communicate with the brain. And all of our tissues and organs communicate through two main modes. One is circulating factors. So a lot of our tissues and organs release signaling factors, maybe they're hormones. Adipose tissue does release a lot of hormones that we call adipokines, and those will travel in our bloodstream up to the brain. A great example from white fat is the hormone leptin. It's telling our brain essentially how much fuel are we storing and can we eat less or do we need to eat more, etc. But those are slow. Those are very slow modes of communication. So when we need a rapid response, like you alluded to, when we're cold, it's an acute stressor, we need to shift our metabolism to provide fuel. The nervous system is the best way to do it. So the nervous system provides incredibly rapid and targeted communication. So our brown fat has distinct innervation by the sympathetic nervous system. So when we need to turn on thermogenesis, the central nervous system, the brain and the spinal cord can send out those signals specifically to brown fat, but they also send the signals to white fat because we need fuel released from our white fat to go to brown fat to keep the thermogenesis going. So it's glycogen, you're correct about that. It's muscle response, but it's also this really rapid nervous system response to our white fat, getting the fuel to brown fat so that we can warm ourselves up quickly.
Okay, so the brain talks to the brown fat. It signals the brown fat. Does the brown fat ever talk back?
Yeah, that's a great question. So I can talk a little bit more about white fat in that sense. So we're very interested in the bidirectional neural communication. Yeah. So the sympathetic drive that turns on thermogenesis, turns on heat production, that's going from the brain out to the tissue. But there's a reverse communication by the sensory nerves. And we might hear this usually about the vagus. So the vagus innervates a lot of our internal organs, and 80% of that nerve are sensory, that are coming from the tissues and organs back up to the brain and sending information about the status or what's needed. We don't know a ton about the sensory nerves in brown fat. We don't know if information about heat or vasodilation or fuel utilization. We don't know if that information is getting back. My lab, we're starting in the white fat and looking a lot at the sensory nerves there and what they're sensing and what information they're sending back to the brain. And it's clear that both of those arms of the nervous system in our fat tissue are talking to each other. The sensory nerves talk to the sympathetic and vice versa. So there's communication, but we need to learn more. We really don't know a ton about how that's working and how that also incorporates these longer term circulating signals like the hormones and other factors that can be released from the tissue. But you alluded earlier that brown fat is much more densely innervated than white fat. And that is true. It has much more of a sympathetic nerve supply than white fat. But the nerves in the tissue are just part of the story because the nerves are releasing factors, neurotransmitters, neuropeptides that can move in the tissue. So part of it is how densely innervated are they, but it's also what are they releasing and how much are the cells in the tissue seeing those nerve signaling factors.
Will you tell me more about that? Because I've read that increased levels of brown fat in an adult human is associated with neuroprotective factors. It could be FGF21. It could be, if I'm remembering this correctly, RMB3, or it could be brain-derived neuroprotective factor, that all of these things associated with increased levels of brown fat are good for protecting, repairing the brain, even reversing cognitive decline. And perhaps you can help us sort that out a little bit.
Yeah, so I think all of our tissues and organs, when they're working in a healthy way, are secreting things that keep the rest of the body informed and potentially also healthy. So I think that's not unique to brown fat. I think that's, all of our organs and tissues, when they work right, are communicating with other parts of the body. But yes, brown fat does secrete things. I actually am the one way back who coined the term badokines. So our white fat releases adipokines, our brown fat releases badokines. So these range from lipids to hormones to other signaling factors, like you said, growth factors, nerve growth factors that can either act locally or they can potentially enter the circulation and get up to the brain. But I think a lot of it, and again, more research is needed here. This is just my sort of informed speculation. But when brown fat is really active, it's metabolically healthy. So it's removing excess fats and sugars from our bloodstream, which we know can be bad for the body. But it's also potentially reducing inflammation. And we know a lot of the protective effects, neuroprotective effects of different interventions, or it could be, these new incretin agonist drugs like Ozempic. These are also reducing inflammation and potentially causing some of these benefits to protect against the risk for Alzheimer's. So I think it's a complex physiology, but I think in general, when our body is working correctly, when our tissues and organs are functioning optimally, the whole system benefits, including the brain. So that's sort of my take on that.
I'm quoted when I'm on podcasts as reminding people that brown fat is a secretory organ. And I typically say an essential secretory organ. But I guess you go back 15, 20 years, medical doctors thought there was no such thing as brown fat in an adult human body. They didn't realize that it can play this critical role in regulating metabolism and protecting the brain. Then we have sort of this revelation from the PET scans of cancer patients saying, what are these symmetrical depots that are hitting positive on our PET scan? They must be brown fat. Nevertheless, 95% of the adult American population, we're talking over the age of, say, 45, has zero detectable brown fat. What can happen to a body that no longer has what I'm calling this essential secretory organ?
Yes. So great question. So a couple things. We can only see what we can measure, right? So essentially what we're measuring is active brown fat. We're measuring uptake of a glucose analog that we can see in a scan, like a PET scan, or sometimes now a lipid analog, which brown fat also uses a lot of lipid. So we have to be able to measure something to see it in those medical scans. So I'm not convinced necessarily that the people who look like they don't have it actually don't have it. They might just have lower activity. So for example, the majority of brown fat cells in the human body are not in these distinct depots that would light up on a scan. The majority are what we call beige. So sort of the tan colored fat cells. They're not brown and they're not white. but they live interspersed in our white fat depots when the tissue is, stimulated. So for example, people with chronic cold exposure will develop more of these beige adipocytes in your white fat that contribute to helping keep your white fat healthier. And they have a similar function to brown fat. So they're designed in a way, their mitochondria are designed in a way to produce heat, and they become more metabolically healthy and have a similar secretory profile. So I'm not sure that someone who gets a scan and shows no brown fat, I'm not sure that means they have nothing. I'm not convinced that means they don't have any badokines moving around in their circulation. I think it's just, we might not have an ability to really see it on that type of medical scan, especially if there are these cells dispersed in our white fat. They're just not going to light up.
Okay, this is important to acknowledge that when researchers at the Sloan Kettering Institute go back and look at 10,000 PET scans, those scans were not conducted under conditions intended to reveal brown fat. The brown fat that showed up on those images was accidental. Maybe the instrument room is a little cold and the subject is wearing one of those blue hospital gowns, and for that reason they might accidentally activate the brown fat and it shows up. That 95% statistic might overestimate the extent to which the population has a deficit. Nevertheless, we know that people who get regular cold exposure, maybe they're an ice bather like I am, or maybe they work an outdoor job in a northern climate. They could be a garbage man or a lumberjack or whatever takes them out into the cold. Those people have brown fat that is located in places, when you know where to look, reliable locations in the body. We know that the more cold you get, the more brown fat you have. Wouldn't there be health differences between those people that have more brown fat and those people that are, I'm going to say, just getting by with the beige fat and the small levels of badokines that beige fat might produce?
Potentially, but I think, isolating that single variable might be challenging. So I think the examples you gave of people with chronic cold stimulation might also be more physically active. They might also have a better diet. They might have a different socioeconomic status. So I think in humans, especially, unraveling these different factors can be complicated. And metabolic health, we always describe it as sort of a scale, with two sides that need to balance. We're taking in energy, and that's a hugely complex system to begin with. Food choice, food availability, how are we storing it? Are we storing it in the right places? And then there's all the energy expenditure side, which is, yes, brown fat for sure, but also the efficiency of our muscles, and et cetera. So it's a complex system. Brown fat is a very important part of it. And secretory factors from any tissue and organ, I think, are also critical to communicate to other tissues and organs. But I don't think we have the data yet to isolate just the brown fat stimulation in those scenarios, because cold does a lot to the body. We're sensing cold on the sensory nerves on our skin. That information is going to the brain. And yes, brown fat gets activated, and that's a great outcome because that's going to build more brown fat if the brain thinks we need more. But a lot of other things are happening at the same time that could be conferring those benefits.
Let's talk a little bit about, at the undergraduate level, some of the vocabulary. I'm an engineer, and so, I build machines. And the difference between triglycerides, fatty acids, lipids, cholesterol, if you don't pay careful attention, you might think these are all synonymous terms. I know they're not, although they might be nested in some cases. Would you mind giving us a short introduction into the difference between these terms that get thrown around all the time?
Yes, absolutely. And actually, I used to teach a course for undergraduates and master's students at Harvard Extension School, and this was one of the slides I would give. So first of all, let's start with the term fat, right? So fat, when we use it in the lay use, could mean obese. It could mean our fat tissue, which we call adipose tissue, or it could mean lipids that we're taking in our diet. So the word fat really doesn't tell us much. But if we move away from that, so the tissue itself is adipose tissue. Having more of it is adiposity or can spill over into obesity. But the lipids that we're taking in our diet are a huge, broad range of categories. So we have signaling lipids that can move around the body and tell our cells different things. We have structural lipids. So the membranes of all our cells are made up of polyunsaturated fatty acids, or PUFAs. And then we have storage lipids, which is triglyceride mainly. So in our fat tissue, which looks very fatty, it's storing mostly this triglyceride. And the triglyceride is a glycerol backbone with 3 fatty acids attached to it. And those can be released. So when you're talking about brown fat needing fuel, what's typically happening is our white fat gets a signal, break apart those triglycerides, send those fatty acids out up in the circulation to our brown fat so that it can use those. So that's sort of the broad strokes. Cholesterol is its own category. It is a lipid, but it's used for different things in the body than just fuel. It's, so there's some cholesterol in our brain that's structural, for example. But triglyceride is the main fuel source, and it's made up of fatty acids that can be released when needed or stored when not needed.
So a triglyceride groups together 3 fatty acids, holds them together with a glycerol molecule, and it can release these fatty acids when they're ready to be metabolized or used in some other capacity.
That's right.
That's helpful. I've also read that the cell membranes and the organelle membranes, for example, the mitochondria, are made of phospholipids. Mm-hmm. And the building blocks of those phospholipids must come from our diet. Mm-hmm. Will you tell us more about fat, not as an energy source, but as a material building block for the body? In particular, you mentioned the brain contains cholesterol as a material, as like the infrastructure of the brain. And Cunnane tells me it's made of these fatty acids like DHA and AA. I think one of the things that people overlook is that the fat they're eating is not just an energy source, sort of a dense calorie source, but it's also what their brain and their membranes are made of.
Exactly. And I think we've learned, since the 1980s, low-fat diets did not do the things we thought it would do for health. And I was actually just at a scientific talk yesterday showing that, in terms of fertility and the relationship of fertility to obesity, having a low-fat diet puts people at higher risk. It's actually not good to have a low-fat diet, but choosing our fats is really important. And the fats that make up membranes, like you mentioned, the phospholipids, those are PUFAs. Those are polyunsaturated fatty acids. They're essential fatty acids. We cannot make them in the body. We have to take them in through diet, and they are vegetable derived sources. So they're not coming from animal fats for the most part. So yes, the composition of fats in our diet, the lipids that we're taking in, make a huge difference for the function of our tissues and organs. And this is completely independent from what we're storing in our fat tissue that can lead to obesity. These are very different lipids. And there are some of these essential fatty acids, as you mentioned, DHA and EPA, which are essential for the brain, essential for brain health and brain development.
So the campaign that maybe reached its zenith in the 80s for low-fat diets was really harming American health?
Yes. I mean, you look at low-fat products, low-fat yogurt's a great example. You take out the fat, it doesn't taste good anymore. So our brains love eating, the person who spoke yesterday said our brains love the taste of palmitate. So this is one of the fats that we take in. It tastes good to us because we need it. But the fact is that you take the fat out of a product, it no longer tastes good. So you have to add a lot of things like salt and sugar, and we know what those do for our health. So it's not necessarily that a low-fat diet is bad on its own, but a lot of the processed food products that take out the fat are removing flavor. Low-fat milk, they have to often add things to change the texture. So sometimes it's flavor, sometimes it's texture. But yes, the low-fat food movement has not proven any benefit.
No wonder the Olean potato chip fell flat on its face, and good riddance to it. But now it's these GLP-1s that are all the rage. And they're popular because people find them effective for curbing appetite and losing weight. Will you tell us a little bit about what's different? Olean was a fat that you could fry a potato in, but you couldn't digest. And a GLP-1 is a signaling molecule. That is, it's changing the way your body thinks it's supposed to work. Perhaps you could tell us a little bit about that.
Yeah, so GLP-1 is, again, a hormone. It's released from some neurons in the brain, but it's also released from our gut after a meal. And it does lots of healthy things after a meal, but it's a really rapidly degraded signal. It goes away in 2 minutes. So because research for decades had shown that it's a beneficial hormone in the body and that it can reduce appetite, pharmaceutical companies worked diligently to try to make a way for it to hang out in the body longer. And that's what these incretin drugs like Ozempic and all these GLP-1s are doing, is just giving you a longer acting GLP-1 signal. And GLP-1 is very effective in the brain at reducing appetite, but its receptors are all around the body. So they're finding now that it does all sorts of beneficial things for the cardiovascular system. Now they're showing benefits for the risk of developing Alzheimer's, which is tightly linked with metabolic disease as well. So this is a really hot time for GLP-1 drugs. But I will say that in the obesity field, a lot of people go off these drugs for various reasons. It's hard to get them covered by insurance. There's side effects that can be very unpleasant, like nausea and diarrhea and things like that. So it does depend. If they work for someone, great. I think they're showing a lot of great benefits, but they might not work. And bariatric surgery is still a great option. Lifestyle, if it works for you, that's also fantastic. But decades and decades of work have shown that that's not hugely effective. Our body really fights weight loss. And then also, I think in your field, if we're driving energy expenditure, that's great because we're burning fat. But the problem with that can be that we're then increasing appetite, because as soon as our brain senses that we're losing our fat, even if we need to lose our fat, it's going to protect that and tell us to eat more. So these medications that make us eat less are really the most effective therapy right now.
GLP-1, though, is not the only hormone that regulates appetite. You mentioned leptin earlier, and I remember Jack Kruse went off on leptin. This might be 10 years ago. He was talking about leptin resistance and the way that leptin controls appetite, and he was advocating for cold exposure as improving the receptivity of your body to leptin. I thought that was interesting, but we're not done. There's also ghrelin. There's also insulin. What happens to the energy in your body? And now there's a confusing lexicon of these hormonal terms that regulate appetite and metabolism for people who are, maybe they don't want to be on a pharmaceutical for the rest of their life. Maybe they've accomplished a lot on GLP-1 and they're thinking, well, how would I get off of this? And what are the other hormones that I might concern myself with?
Yes. So I think it's interesting when you look at all the different signals in the body that control appetite. It really tells you how important it is. We're not relying on one signal. We have all these redundant pathways. So GLP-1s are one signal, and those work quite well. Leptin is tough, and so is insulin, as you mentioned, because when you're obese or you have diabetes, you're resistant to those signals. So they haven't proven to be good therapies, but people are still working on leptin and whether or not we can adjust the body's receptivity to leptin. But in general, when people develop metabolic diseases, they've lost the ability to respond to leptin and insulin. And that's really the problem. And we don't fully understand those mechanisms yet. But yes, what's interesting about the GLP-1 medications, they're very effective. But just like any therapy for any disease, if you stop taking it, you lose the beneficial effect. So your disease doesn't go away when you stop taking a medication. These are not permanent cures. Bariatric surgery, for those who are responsive, that can be a permanent cure. But obesity is a disease. And people that have a bigger than average appetite drive, this is built into them. And so it's going to come back if you go off the medications.
There is a psychology of appetite, not just a physiology of it. Are you familiar with the milkshake study?
I think I am. Remind me though, that there's several studies.
Notorious at this point. But one of the co-authors was at ASU, where my faculty position is. Now, I've never met them. I've never talked to them, but I find the study fascinating because they had two identical drinks. So these would be like protein drinks. They told one group that it was a dessert shake, and they told the other group that it was a diet shake, and they were identical. The people who got the dessert shake reported feeling more full and more satisfied. And the people who got the so-called diet shake were like, oh, well, now I still kind of need something. I still have an appetite. It had nothing to do with the physiology. It had only to do with the framing of the experience of drinking the shake. And they
Yes, and
measured ghrelin.
Yes, and yes, so ghrelin goes up in the gut after we eat a meal, so that is another appetite regular. Sorry, it goes up before we eat a meal, so that's another appetite regulatory signal that's telling us we're hungry, and it's we train that signal. So ghrelin goes up when we anticipate a meal, which we can notice when we travel and have jet lag, and we are like, oh, I'm hungry for lunch now because in my home time zone I should be having lunch. But yes, so what you're hitting on is this really complex neuroscience of appetite and food intake. So the region of the brain I study, the hypothalamus, this is really homeostatic food intake. So this is the food intake that keeps us alive. So we've lost some fat. We just ran a marathon. We've woken up in the morning after fasting. We need to eat. That's the signal that our hypothalamus is receiving and coordinating the food intake response. But the rest of the brain is responding to the look of food, the smell of food, the taste, the mouthfeel, and also, to your point, the psychology, what we think it should be doing for us or what we anticipate. So it is very complex. I'm not in this research area. I have a lot of colleagues who do great work in this area. These incretin drugs like GLP-1s, they think are affecting some of these food responses. But yes, it's more than just do we need calories. There's a huge, societal, psychological component of our nervous system that's involved.
Was talking to Holly Wyatt at the University of Alabama, Birmingham, and she said, I think the term was food noise, that there's sort of a background chatter that happens in our brain thinking about food, or we have thoughts that stray involuntarily to food, and that the GLP-1 helps quiet some of that food noise in our brain. And that is enough to take the edge off of appetite sometimes.
That's right. And the GLP-1s are also good at regulating our glucose and insulin system. And that's a huge part, as you mentioned, that's also leading to the hunger. But that food noise and our, how much we eat at a meal, how much we're responding to all these external food signals, these are highly variable between people and highly genetic. So, telling someone that they need to just have willpower and eat less and not think about food in the middle of the day when the person next to them is thinking about it, that's easier said than done. And I think we've really proven that this is biology, this is physiology. And when the system isn't working properly and you get to the point that you've increased adiposity and now you're getting diseases like obesity and diabetes, this is a pathophysiology. This is a disease state.
There is a relationship that you're investigating. If I'm reading your Scholar Google profile correctly, there is a relationship between sex hormones and fat tissue. And
that's right.
I mean, we've known for a long time that testosterone goes down as obesity goes up. But I think you're looking at it in a more detailed way. Will you tell us about some of the work you're doing there?
Yeah. So some of this starts with just looking at the distribution of fat in the body and the way people gain weight when they're becoming overweight and obese. You can see differences between men and women. So it used to be called the apple and pear shape. That's kind of fallen out of favor, but we've introduced the concept of subcutaneous versus visceral fat tissue, and women build more subcutaneous, which can be healthier. And men, when they become obese, tend to build more of the visceral fat, which can lead to more diabetes, for example. So there are already sex differences in where we store fat and how we store it. But what's interesting is our fat tissue is a sex hormone endocrine organ, and it actually helps convert androgens into estrogens. So when men have more adipose tissue, they will be making more estrogen, and estrogen and testosterone talk to each other. But then we get into menopause. And for women, I think this is a really interesting metabolic period of life, and maybe a time when people are starting to use some of these incretin treatments. But as we go through menopause and lose our estrogens, we start to gain more visceral adiposity, so the unhealthy fat. And this is really what's driving the increased risk for metabolic disease as women age. And you can see it when women are put on hormone therapies, that starts to reverse. So there is a direct relationship to our hormone levels and then how our fat responds, but also our fat can make hormones like estrogen.
Marty Macari pointed this out in his Blind Spots book before he was appointed the head of the FDA. And he was very positive on hormone replacement therapy for menopausal women. What you're saying reminds me of what he's been talking about, but I think there's still a lot of opposition.
Yeah, it's getting better. So, it stems from a lot of older studies that were done on women getting hormone therapies after they had gone through menopause, and they did see some cardiovascular risks. And there still can be, some cardiovascular risks. You have to work with your doctor closely on what hormones you're getting and at what levels and combinations and things like that. But newer data, arguably better designed studies that have been done more recently, show that if you give hormone therapies more in the perimenopause phase, so before you feel it fully gone through menopause, then you're really seeing the benefits. So again, women's health is an area that's underfunded. We need more research. We really don't understand how to optimally deliver these hormones, especially testosterone, at midlife to women. So hopefully we'll see more evolution in that space.
Well, I want to see if I can thread a few of these themes together now, because all sex hormone synthesis originates in the mitochondria. It is the inner membrane of the mitochondria where cholesterol is converted to pregnenolone. And then all the other sex hormones are downstream of pregnenolone. What we've established so far is that brown fat is mitochondria rich. Mm-hmm. My testosterone jumped like through the roof when I started doing ice baths every day. And now I have a number of case studies, men and women, who started a regular practice of cold plunge therapy. We know that this activates their brown fat. We know that this will recruit new brown fat into their body, and that brown fat is dense in mitochondria. Then I talked to Martin Picard at Columbia University, and he pointed out that mitochondria will travel through the body. The mitochondria synthesized in the brown fat don't necessarily stay there. There was one study that he pointed me towards showing that mitochondria can be released from fat tissue and travel to other organs inside the body. Those organs might be in a man, perhaps the gonads, where most of his testosterone is synthesized. But in women, it's not just the ovaries. It's also the adrenal glands, the skin cells, and fat cells that are capable of producing pregnenolone. And so now you can see how I'm trying to tie the mitochondria in the brown fat, the secretory role of adipose tissue, and the sex hormones together, and ask you if we're on the right track here.
Yeah, I think that's a really exciting area of research. So yes, mitochondria can leave a cell and move, and that's the really interesting concept, because the mitochondria in brown fat are unique. Our muscle and our liver are also very dense in mitochondria, but those don't have uncoupling protein 1, which is what allows those mitochondria in brown fat to make heat and be so metabolically healthy. So those are unique mitochondria. But also we're learning mitochondria are moving around inside of our other cells. So if you think about these neurons, these nerve cells that are innervating our fat, some of these have very long axons, and the mitochondria need to move that entire distance. So keeping our nervous system healthy so that mitochondria can move and allow a neurotransmitter to be released, for example. This is another area that I think we really need to do more work. But yes, mitochondria are fascinating organelles in the body, and they're very bioactive, and we need to learn more about what they're doing within cells and also communicating between different tissues and organs.
The idea that mitochondria are mobile, to me, is a revelation. But I didn't realize that some mitochondria had the uncoupling proteins that you would find in brown fat, and others didn't. How malleable are these mitochondria?
Yeah, interesting. So mitochondria are very dynamic organelles. So they can break apart. A mitochondria can break apart and make multiple mitochondria. We call that fission. Or smaller mitochondria can come together and make a larger mitochondria. Or as we're growing more brown adipocytes, right? So we could be making our brown fat bigger, or we could be making more beige cells in our white fat. Those both need mitogenesis, so making more mitochondria. So they're highly dynamic organs. But this is getting into this idea of plasticity, which is really a big favorite topic of mine, that our adipose tissues in general are highly plastic, our mitochondria are highly plastic, and our nervous system can be highly plastic. So meaning dynamic, it can remodel, it can respond to signals and change how it functions. And to me, across what we study, which is the nervous system and our fat tissue and how those need to stay healthy so we don't get metabolic disease, for us, the theme that's running through all of it is maintaining these plasticity responses. So can we make more mitochondria with the right expression of things like UCP1 if needed? Can our nervous system regenerate? Can our adipose tissue expand and store the fuel that we have and not let it spill over to our heart and our muscle, et cetera? To me, those plasticity systems, those mechanisms are really the key to health and health as we age.
Can our nervous system regenerate?
Great question. So the peripheral nervous system is very good at it. And unlike the brain is not very good at making new neurons. But the support cells, the glia that help the neurons function, are highly plastic, and then those can turn over in our life. But yes, the nerves in our white and brown fat, if you're chronically exposed to cold, they will grow out and branch. We will increase the innervation of our fat tissue with chronic cold exposure. And to us, it's one of the best examples of peripheral nerve plasticity because it's just so dynamic. We can see it in the mice very easily. We can look at the molecular processes underlying it. And then if you reverse that, if you rewarm an animal, those neurons will grow back. And then you put them in cold again, they'll grow out and branch and increase density. It's just a very responsive element of the peripheral nervous system, looking at a demand like heat production and fuel utilization for that heat production, and then responding to it. It's really incredible.
In some disorders like multiple sclerosis, there is damage to the myelin sheath. And some people are telling me that they believe that myelin sheath damage can be repaired. Have you witnessed this or researched this? Do you have an opinion on this?
So I'll give the disclaimer: I'm not an MS researcher, but we are interested in the cells that myelinate our peripheral nerves. They're called Schwann cells. So these are unique cells that use a lot of specialized fats, actually, and build this fatty sheath, this myelin sheath along axons, so that the neuronal signals, which are electrical in nature, it's like insulation on a wire. You need that fatty layer to transmit these nerve electrical signals. And so when that breaks down, and it could be MS, or it could be the neuropathies that we look at. So neuropathies that happen with obesity or diabetes or aging also have a loss of the myelin. And so then your neuron can't function properly. So we're very interested in whether we can coax and sort of convince these Schwann cells to become more plastic, to regrow the myelin sheath, and to help improve the function of nerves. So I do think it's possible. I think we're really at sort of the forefront of this type of research. I think we know a lot about how these cells function, but there haven't yet been very effective therapies put into place. But that pipeline of therapy development is also very long. So we'll see what's coming, but I'm very optimistic.
This is extraordinary, thinking about fat as a therapy. Now, that's a little bit of a leap from where we are now, but fat has been so demonized, both in our diet through these public campaigns of like, you got to go low fat, and in the understanding of our own metabolism and our physiology, where we think about fat as a bad thing. And you're pointing out lots of different roles that fat plays in the body that protect us and keep us healthy.
Well, yeah, because the term fat, as we've covered, is not a catch-all, and it shouldn't be a catch-all. There's thousands of types of lipids in our diet and in our body that are carrying out very different functions. And some of them, if we have too much, can be unhealthy, of course. So it's really about what types of fat do we have and what ratios. And so I think we're still learning more about what's important there and which certain fats or certain lipids might be therapeutic, and whether that's situational, right? So maybe our nerves need one type of fat to be beneficial to turn on this myelination, but that would not be good for our liver, right? I'm just speculating, but I think this is a highly complex system. And when we oversimplify and say, let's just reduce our fat intake, we've shown that that's not effective because it's too complex to make a broad generalization like that.
Well, I'm already eating the full fat yogurt. I love my whole milk. I'm very into the dairy products. But suppose I'm convinced, Kristy, what are the fats, the healthy fats that I should have in my diet so I can keep my brain and my nervous system and my phospholipids operating well?
Yeah, I mean, the key is really a balance. And this is, an area of nutritional science that, again, we're still trying to figure out what the right balance is. And does it depend on who you are and your genetics? And should there be, some personalized approaches? But in general, the saturated fats that are coming from things like coconut oil or animal products that are solid at room temperature, we need some of those, but having too much, we know, is not good. Trans fats, which are the more processed food fats, definitely not good for metabolic health. But the polyunsaturated fats, the ones that help give us those essential fatty acids, the ones that are plant derived, the ones that our cell membranes need, really, really critical and tend to be too low in our diets. And a component of that also, one of the polyunsaturated types of fats is omega-3, which is, can come from seeds and nuts and fish and things like that. And we really, especially as Americans, are getting too little of those polyunsaturated fats, the omega-3s and the omega-6s, and we need to be getting more food-derived sources of those.
I've heard that from many sources. I think that there's a blood test you can get that will measure your omega-6 to omega-3 ratio. At least I have this recollection of reading it on one of my reports and being shocked by how I was so far out of whack. I had to make some changes in my diet to minimize sources of omega-6 because I became concerned that I wasn't giving my body enough of the omega-3. And I see from you nodding your head that you think probably a lot of Americans, that's a typical situation where their omega-6 to omega-3 intake is out of whack and they should reduce omega-6 and increase the omega-3.
Potentially, yeah. I mean, omega-6s are good. I think they get vilified in the lay press, but we need omega-6. There's essential fatty acids in there that we need. But yes, I think it's easier with the typical diets that we eat. It's easier to get omega-6, and we have to work a little harder and be a little more intentional and aware to get enough omega-3. So they can be out of whack for sure.
Kristy, where is your research going next? When I look at the papers that you've published, I know that what I'm reading could be 2 or 3 years old just because of the way peer review and revision and publication works. Where are you going now that is keeping your curiosity active?
Yeah, great question. And it ties back to a lot of the themes we've talked about today. So my favorite area right now in our lab is looking at how our dietary fats affect our nerve health. And if damage to the nerves with an unhealthy high-fat diet that's not well balanced is the initial step that might cause metabolic disease. So we know that peripheral nerves die. This is neuropathy. It happens with obesity. It happens with prediabetes, and it definitely happens with diabetes. It also happens when we age. And so stopping that, but then also repairing it is a key focus of ours right now, because we believe when the nerves are healthy and they're helping to keep our fat tissue healthy, then the whole system can function properly. So really this idea of the connection between nervous system health and metabolic health.
Okay, if I understand this correctly, neuropathy is a death of the peripheral nerves. And one of the things that you mentioned earlier is that peripheral nerves can be restored, that you can make changes, could be in the diet, but it could be in other lifestyle factors that reverse this neuropathy by regrowing peripheral nerves. That sounds very
Exactly. So in theory, it's possible, but I will say that neuropathies, which affect 30 million Americans right now and 130 million or more worldwide, there is no cure. So when people have neuropathy that's so severe that they're getting pain, they're getting numbness and tingling, they can't cure that right now. So lifestyle is not enough in those states. Can lifestyle be protective? Definitely. But when you get to that point of developing a neuropathy, there is no treatment currently. But because we know the peripheral nerves uniquely are so plastic, they're so regenerative in a healthy state, we're trying to learn more about those processes, those molecules that are involved, and whether they could be a novel therapy in the future. And lots of people are working in this space. But I think we will see one in the next couple decades, a treatment for neuropathy. But right now, I think prevention, keeping your nervous system healthy as much as you can. And part of that is eating healthy fats, we think. We think unhealthy fats can be neurotoxic, not just to the peripheral nerves, but probably also to the brain. And that those would be one of potentially many contributing factors to neurodegeneration.
Well, I think in 1 hour, we managed to reverse decades of misconceptions about fat. It's quite an accomplishment. I'm looking forward to the papers that are going to come out in the next year or two. And I can't thank you enough for joining me.
Well, thank you very much. And thank you for covering this and helping scientists like me be able to share what we do with the broader public. I think that's more important now than ever, and for helping people understand the terminology, which can really become confusing even for us sometimes as scientists. So thank you so much for what you're doing.